We're going to keep going with the organelles of a cell, specifically a eukaryotic cell. In the first lecture on the organelles, we talked about the nucleus and the nucleolus. We talked about the ribosomes. Now we're going to get into some more of these organelles. And this stuff should sound familiar to you. My hope is that as I explain it, you're not only remembering, but you're building on that. And hopefully I want you to see how these things all work together. Okay, so the next one's called the endoplasmic reticulum, often called ER for short. So we're going to refer to it as ER, but it's endoplasmic reticulum. And what this is essentially, these are the passageways that it's how things get around the cell. So the endoplasmic reticulum actually is continuous with the nucleus, so it's attached to the nucleus, and it is how information gets to the ribosomes in order to make proteins, and we're going to talk more about that in a minute. But basically, these are passageways that allow for proteins and other materials to transport around the cell. And there's really two different types, and the two different types are called the smooth ER and the rough ER. And the smooth ER is going to be really, the main job of that is to transport proteins and finish them up. And the rough ER is the reason why it's rough is because it's covered in ribosomes. So that's an easy way to remember it. Rough ribosomes, RR. And it's covered in ribosomes, and this is where proteins get made. So this next image will hopefully, you'll see the difference between them. So essentially this would be the nucleus here. So as you can see, the nucleus is connected to the endoplasmic reticulum, and you have your rough endoplasmic reticulum, which is going to be closer to the nucleus. Here, I'll move my head over here so I'm not blocking it. And that is where the ribosomes are, and this is where proteins get made, and then as they get made, they get sent out, and that goes through the smooth endoplasmic reticulum. So essentially, think of this as a factory. And we're going to talk more about this in a minute when I get to the Golgi bodies. But the nucleus has the directions on how to make the proteins. The information is sent to the ribosomes through the rough ER to make the actual proteins. Then when the proteins are made, they get sent through the smooth ER out to the cell. And the next structure is called the Golgi body. Just to go back to this actually. I'm trying, because you guys are filling in some information. The endoplasmic reticulum are going to be found in plant and animal cells, but not in bacterial cells. Okay, the next structure is called the Golgi body or the Golgi apparatus. They're interchangeable. And this is where we actually package proteins to leave the cell. Here's the thing. Most of your cells, their jobs are to make proteins, and a lot of those proteins get used outside of the cell. So for example, we have glands, and those glands produce hormones. For example, adrenal glands produce adrenaline. We have glands in our hair called sebaceous glands that produce oil. That's why your hair gets oily after a few days of not washing it. And what ends up happening is before these proteins can leave the cell, before what gets made can leave the cell, it goes through something called the Golgi body or the Golgi apparatus. And it basically surrounds these particles, these molecules, in something called a vesicle. And it's basically it's a membrane that surrounds it. If you want to send out a letter, I know nobody mails anything anymore, but let's say you wanted to write your grandmother a letter. She likes getting cards. So you write up a nice little card, and then you just bring it outside and put it in the mailbox. It's not going to go anywhere because you didn't put an envelope on it with the address. And if you did put the address right on the card, it could get damaged because the envelope isn't there to protect it. So think of the Golgi body as being the part of the cell that protects the proteins before they leave the cell. So it's like the mail. It's like the mail. Basically, it packages these proteins so they can leave the cell and go where they need to go. And basically, you can think of it as putting on the finishing touches. And there's two sides to it, and we don't need to know this detail, but the cis phase and the trans phase, that is just which side leaves the cell. So basically, proteins that are getting made leave the smooth endoplasmic reticulum, they go into the Golgi body, and then the Golgi body packages them, and then they leave the cell and through the cell membrane. So the Golgi body is going to be found in plant and animal cells. Lysosomes. Lysosomes are very important because here's the problem. Things stop working at a certain point, right? So like, for example, you might have an organelle like a ribosome that doesn't work anymore. It stops functioning correctly. And lysosomes are there to clean everything up and get rid of it. So lysosomes have digestive enzymes in there. And digestive enzymes, as you know, enzymes are proteins that break things down. So the job of these lysosomes is to digest food partially, right? To break food down. These cells need to get nutrients. So that's one job of these lysosomes. And they're also responsible for destroying and getting rid of organelles that don't work anymore, just to dissolve them. So if you take a look here at this diagram, you can see this just shows food coming into a cell, and then a lysosome coming from the Golgi body, right? So the Golgi body does its job and packages the enzyme into a lysosome, and then the lysosome is going to put its digestive juices into the food and digest it. Also, it could be used to break down organelles that don't work anymore. So let's say it's a mitochondria that does not work anymore. The lysosome is going to surround it and digest it and get rid of it. So think of the lysosomes. Lyse means to cut, right? Hydrolysis means to cut with water. Lysosome means to cut or break things down. So it breaks down food and waste products, essentially. That's the job of our lysosomes. I'm going to keep moving. Oh, they're also found in plants and in animals. Vacuoles. So vacuoles are basically, they're very similar to lysosomes, but instead of being full of digestive enzymes, they're just storage. That's what they are. They're there for storage. Often they store water. That's one of the main things they store. They store food particles. So vacuoles are going to be found throughout the cell. They're small membrane-bound organelles, and their job is they are just there for storage. Plants have something special, though. Plants have something called a central vacuole. They have one very large vacuole that exists right in the middle of the plant cell. Animal cells do not have this. So, if you want to, one of the big differences between plant and animal cells is that plant cells have a large central vacuole, and that is responsible for storage. And the reason why plants have a large central vacuole is because that's where they store water. And guess what? Plants can't do. Plants can't get up and move if there's no water around. So if a plant exists in an area where it doesn't get rain, it needs to survive throughout that time where it's not getting rain. So it stores that water in these central vacuoles. So just make sure you understand that plants have that. A great example would be like cactus. Plants like that are in very dry areas, they're going to have very large vacuoles. So that way they can store water. So when it's not available, they have it. We don't need that because if we don't have water around us, we can move. We can go to where the water is. Plants don't have that ability. So they have a large central vacuole. So I'm going to finish up here just by going over the few that I covered today, just how they're connected to each other. So let me just explain that. So I just want to, like I said, review a little bit about what I talked about today. And there's a handful of organelles that work together. So let's say I have a cell here. So it's a cell. Now we have our nucleus. Inside of our nucleus, we have our DNA. So I'm just going to make that. That's going to be our chromatin, all jumbled up in there, right? And then we have our endoplasmic reticulum, which is kind of attached to that. And on that, we have our ribosomes, right? So we're going to put our ribosomes on there. This is our rough ER. Okay. And then the next thing is the smooth ER is attached to that. So I'll just kind of extend that on to that. So this will be your smooth endoplasmic reticulum, which again is just there for transport. And then finally, we have our Golgi bodies. Make that, whatever. This color here. So how are these things all related to each other? Now, let's say I wanted to make a protein. What would end up happening is the DNA would supply the information to the ribosomes. So the information would get carried to the ribosomes, and I'll talk more about how that happens a little later on, and we make our protein. So the instructions are read, and so now we have our protein. That's our protein there. And what's going to happen now is we're going to move that protein through. So it's going to travel through the rough endoplasmic reticulum. It's going to travel through the smooth endoplasmic reticulum. And now our protein got made. And now let's say it's going to get used outside of the cell. What's going to happen is it's going to then pass through the Golgi body. And what the Golgi body is going to do is it's going to surround it in a protective coating. So let's, whatever. Let me get this color. So it's going to surround it in a protective coating, and then that protective coating is going to keep that protected while it leaves the cell and goes somewhere else. So that's essentially how those things are together. So you got your DNA in the nucleus, the rough endoplasmic reticulum has the ribosomes attached, the smooth endoplasmic reticulum is for transport, and then our Golgi body's there to protect that protein as it leaves the cell. Thank you very much.